Alrighty, everybody. I'm really excited to introduce Ben Meinen, our speaker group. So Ben has been in my group at IPI for only about 6 months now, I think, or so. So this is very much a work in progress that Ben's going to share tonight. But yeah, I'm really excited for how far he's come in such a short period of time. So Ben, please take it away.
Well, thank you for the kind introduction and welcome everyone. Yeah, I'm excited to share my project, and I will talk about rational de novo design of agonists and antagonists for family B G protein coupled receptors. And so as an outline, first going to tell you why we care about family B GPCRs and why they are interesting and difficult to address drug targets. Walk you through my computational de novo design strategy and the process, how I decided to filter and select some designs, and then how I made and tested them in vivo, or in the test tube, I guess. So first, G protein coupled receptors are integral membrane proteins, and they can be activated by agonists and or for a range of stimuli, can activate it by light in our eyes, or adrenaline or serotonin in our bodies. And when they're activated, they recruit a G protein, and the G protein can then bind to the receptor and engage secondary messengers, such as cyclic AMP or calcium, that then activate a range of physiological functions. So there are around 800 GPCRs in the human genome, and around 30% of all FDA-approved drugs actually target the GPCR, which makes them a very important class of drug targets. Family B GPCRs are characterized by this large ectodomain shown here in orange. And it's a fairly small family. There are only 14 members, and they're all regulated by hormone peptides. So here's a structure of this family B GPCR bound to one of the hormone peptide structures. They are involved in all range of responses, from diabetes to osteoporosis to anxiety and some cancers. And at the moment, there are only 3 agonists that are clinically approved by the FDA to target these receptors, and there are no antagonists approved. And so it's not that antagonists wouldn't be helpful. It would be very useful to have antagonists for a bunch of these receptors. They just haven't been clinically approved yet, and for some of them, there aren't even any experimentally developed antagonists that can be used to further investigate the receptors. So making agonists and antagonists for all these class B receptors would really help the field to study them in more detail. So that's why we're really interested in doing that. And so I will mainly focus on talking about one of these receptors, the parathyroid hormone receptor 1, short for PTH1R. And so I choose this receptor because the cryo-EM structure is very high resolution and gives us a good input structure. So one shown over here with the peptide bound. It is involved in calcium homeostasis and bone turnover. So it's a drug target in osteoporosis. And it is one of the better characterized class B GPCRs. And just for the terminology, so if I talk about an agonist, I'm referring to a full agonist. So if the agonist is bound to the receptor, it leads to 100% biological response. And an antagonist is a molecule that doesn't result in any response when bound to the receptor. So yeah, our goal is to make agonists and antagonists for all family B GPCRs. So it is challenging for several reasons. One of them is that the natural peptides, so here on the left side, I show you the solution NMR structure of the PTH1 to 34 hormone. They are intrinsically disordered proteins, and they only, when they're bound to the receptor, adopt a regular alpha helical structure. And how the binding occurs, it is thought to occur in 2 steps. The first step is the C-terminal part of the peptide engages with the ectodomain of the receptor and forms a stable complex. And then in the second step, the N-terminal part of the peptide interacts with the transmembrane helices and leads to a conformational change in this helix 4. And so that then recruits a G protein and leads to the signaling downstream. And so if we just do a thought experiment, if we look at this disordered agonist protein, if it binds to the receptor, there's a lot more binding surface that it can attach to, and it engages in a very stable binding. But if we remove these agonist parts, so shown in purple or pink over here, we end up with a shorter, smaller, also disordered peptide, and the binding to the active domain is entropically very disfavorable, so it only leads to weak binding. And so what we really like to do is to inhibit the binding of the agonist by binding in the same binding site as a natural peptide, but without activating the receptor. So we want to do that with Denovo design. And so here again, on the left side, I show you the input structure. So that's the natural peptide, and we separate it in the agonist N-terminal portion and the C-terminal portion. And so our design goal is to attach 2 more helices to the C-terminal part of the peptide. And with these 2 helices, we stabilize the C-terminal peptide. And as an agonist, we want to do that without interfering with the agonist portion. And so if we can successfully make an agonist that activates, we hope that we can convert these agonists by removing the N-terminal agonist part and then stabilizing the C-terminal part in a binding competent conformation to tightly bind to the active domain. And then we have a competitive inhibitor of the binding site. And so we want to do that with miniproteins. And Denovo miniproteins have a lot of advantages. They can combine the affinity and specificity of antibodies with a lot of advantages small molecules have, because they can penetrate more tissues. And in our Denovo design protocol, we have complete control about the properties and activity. We strive to design a stable hydrophobic core, so we can direct folding in the right conformation. And we introduce disulfide bonds in our structure that lead to high thermostability. And so the high stability will hopefully make it possible to store them at room temperature, which makes them a more accessible or equable molecule. And lastly, they are easy to manufacture because we can genetically encode them and change them. So we want to do our design as a requirement-driven design approach. And so the requirements we set for these designs are that we want to have a 3-helix bundle, and this 3-helix bundle should include this C-terminal helix part and stabilize it in a binding competent conformation without interfering with the N-terminal agonist part. And after we have achieved this, we also want to integrate a C-terminal disulfide bond to make it a stable miniprotein. Our design process has 3 steps. So first step is our backbone design, where we use So Anything, which is developed by Frank in our lab, and it's a modular improvement to the sewing code that is published before. And so with this protocol, we attach 2 helices to our starting structure to make our backbones. And I will go more detail in the following slides. So that's just an overview. But after this first step, we select the backbones that fulfill all the requirements we set. And then we introduce a C-terminal disulfide bond, where we use the Hemlock protocol that's also developed by Frank in our lab. And after we have successfully integrated this disulfide, we go to our Rosetta sequence design stage, where we use layer design and fast design to find sequences that can represent our backbone and fold up in the right conformation. And then we select and filter the best ones or the most desirable ones to test them. So sewing. The sewing stands for structural extension with natural fragments. And what we do is we have this pool of millions of helix loop helix structures from the PDB. And we give, in this case, I give it the input structure with these this input helix, represented here in blue, and we sample this pool of fragments to find fragments that align well with our input structure. And when we find such a fragment, we chimerize it together. So we have then 2 helices, and then we can repeat this step with the second helix, and then end up with 3 helices. And after we have 3 helices that fulfill all our criteria, we move them on to our next stage. And in our next stage, we want to engineer a C-terminal disulfide to increase the stability of this scaffold. Also in this case, right, we want to keep a free N-terminus because it's signal, so we don't want to engineer any N-terminal disulfides. And so the Hemlock protocol has also 3 steps. So in the first step, we use so anything again, where we have this pool of fragments with the PDB, but this time it's just a helix and a loop. And we again try to find loops, helices that align well with our third helix and have a long loop. And then we use the disulfide mover in Rosetta to find positions in this attached loop that can form a favorable disulfide bound with any point in our backbone to stabilize the termini. And when we find such a position, we engineer in the disulfide. And then in the last step, we delete any loop residues that are left over. So the cysteine that forms the disulfide is our last residue in the design. And so here I've shown 3 backbones I generated, and I just want to demonstrate that this disulfide protocol leads to a range of different disulfide bonds. So in this, we have a short loop and a linkage between the last helix and the first helix. In the middle, there's a longer loop, but there's the longer loop and the connection is also between the first and the third helix. And then in the last structure, we also have a longer loop, but at this time, the second and the third helix are connected to each other. And so after we got all our backbones that fulfill our requirements, we move them on to our backbone design stage, our sequence design stage, where we want to find sequences that represent them. And so I'm going to share with you what we consider our secret sauce to sequence design. And in our first step, we first define our layers and secondary structures with a residue selector. So we select the residues in helices, shown here in cyan, residues that are at the start of the helix, shown in orange, the helix cap, shown in pink, and then the loops in green, and beta sheets, which in my case are just all helices. And then secondly, we select for the residues by their position. So we select for surface residues, so residues that are pointing outwards of the structure, for core residues that are buried inside the backbone, and then boundary residues that are in between the surface and the core. And so these residue selectors are passed into our design restrictions, where we define which amino acids to design and what operations to do. And so there are 2 kinds of operations: one, residue level operations that direct the packer to do certain things, and then the design restriction for the amino acids. So in this first block, which we use for the design of the monomer backbone protein we're using, we will allow for more aromatic rotamers to be sampled. So this allows us to get a tight core packing of the core residues and all the aromatics. And then in the interface, we additionally to the aromatic residues allow also some more rare rotamers to be packed and designed to just get a very diverse interface. And so in the first layer is the surface layer. So here in the surface layer, we allow mainly charged and hydrophilic residues to be designed. In the boundary layer, we allow charged hydrophilic and also some hydrophobic residues to design. So that's our most promiscuous layer we design. And then in the core layer, we only allow hydrophobic residues to be designed. And then for the helix cap, we only allow the residues that stabilize that, which comes from the Rockland paper a couple years ago. And then that is the block that defines our interface. So as I said, for the interface, we allow some more rotamers, and we allow the interface residues on our receptor to repack to get more different binding conformations for the side chains, which should all be possible, but we don't allow any design of the rest of the receptor. And then the residues in the interface, we allow also a set of hydrophilic and hydrophobic residues to be designed. So after that step and we get our backbones, the question is really, how do I select our designs or full designs? How do we select for these designs which we should order or which we should test? And so I decided to look at a bunch of metrics because I made around 2,500 designs. I personally can't look at 2,500 designs by eye and pick them. So I decided, secondly, with our design protocol, we want to automate as much as possible and have really metrics to look at. So here I'm just plotting some binding energy metrics. So on the x-axis is the DG separate, which is the energy it takes from the miniprotein to be separated from the receptor. So the higher the energy is, the tighter the binding. And on the y-axis, I plotted the binding interface. Also, the larger the interface is, the more residues can interact with each other. So we would assume that designs that fall in this upper left corner are the designs with the most desirable binding. And another binding metric we can look at is shape complementary, also plotted on the y-axis here. And for shape complementary, you can think about if a key fits into a lock. So if you have a key that really doesn't fit into the lock and doesn't have the right shape, we would assume that has a worse shape complementary, so a low value. But if you have the right key that has all the right grooves, it will fit well into the lock. So we will get a high shape complementary. And so we are really interested in the designs that are also here in the upper left corner.
Just a quick question, if I may.
Yes.
How do you fit the designs into the receptor? Do you superpose them with the natural antagonist helix? Or how do you stick it in there? Or how do you start a docking or whatever step you're doing there? I mean, it looked so far as if the designed molecule is designed on its own, and now you have to stick it in the context of the receptor.
So the design molecule is designed by its own, but the receptor is present during all stages. So even in the sewing stage, the receptor is there, so we avoid any clashes that might be occurring for our backbone. So we design in the bound state.
And how do you position it then?
So we use the starting PDB structure where the agonist peptide is bound as the position structure and then superimpose on that. So we don't need to dock it in because we just have the complete structure for this receptor.
Okay, and so one of the helices in the 3-helix bundle has, how does Rosetta know to superpose the correct of the 3 helices with that starting structure?
So we can label our input structure with a residue labeling metric, and then these labels will. So right, if we design it, the number of the numbering of the miniprotein might change, but we can label them in the beginning, and these labels will also already be attached to the right position in the initial helix. So we can always refer back to that label, and that's what we use to position it.
Okay, thanks.
I hope that helped. If anyone has more clarification, happy.
I have a question too while we're here. Would you expect more correlation between the binding shape and the binding energy, or is it expected that they're not very correlated?
That's a good question. I think that you can have for example, a very hydrophobic interface that just excludes a lot of water, but doesn't fit well, and that would still give us a high binding energy because it just takes a lot of energy to disrupt it without having a very good shape complementary. So you kind of want to be at the intersection where it fits well, but also has a good energy. So, okay, next. So, but we are not always, we are interested in designs that bind really well, but we are also interested in having our designs being really stable. So, and a metric to determine how stable our designs are, I looked at residue as an interaction energy metric. And so here I just select residues that are in the core of my protein, and I calculate the full item attractive energy term. So that's the van der Waals energy term for the attractive energy, and gives us an idea how well packed and how basically how much water is excluded and how stable the hydrophobic residues in the core are. And the more negative, the mean. So, and then I basically, after I measure the value for each core residue, I calculate the mean. And so the higher the more negative the mean, means the more stable each residue in the core is. And that is for us a good indicator to see how stabilized the core residues are that hold the protein together. So basically I want to be at the intersection of high binding energy and high core stability to get proteins that are stable, but also bind well. And so to achieve that, I set stringent filter criteria based on what I saw from my plots. And I used some filters for local and global structural cooperativity that we are developing in the lab. Then the core stability I told you about, and then the binding criteria. And these, I guess I have to give credit to Jared and Frank for developing these filters and helping really well with analyzing which designs are good, which designs are bad. And so after I filtered that, I got around 100 designs. And so from these 100 designs, I wanted to order 48. That's a good set to handle and make. And we wanted to make them in mammalian cells. So I filtered out sequences that could be glycosylated, and then I rank ordered them by binding energy. And so from that, these are the final 48 I then ordered. And so I can show them you here. And so we make de novo designs so we can name them however we want. And just giving it PTH1R agonist design seems fairly annoying and boring to me. And I like for sure breaking that quite a bit. So I decided to name all my designs Walter. So we are now working with Walter 1 to 48. And so here are shown these designs. And so again, we did not pick them by eye. They were all determined by our computational metrics. And it's quite interesting to see that even they're all 3-helix bundles and have our C-terminal disulfide, they have a range of crossing angles and different conformations while scoring all pretty similarly. So that made us very excited to test them and see how well we could predict if these are good designs and if we can make functional designs. And so So next up, I wanted to characterize the designs. And so we choose to do that in mammalian cells because our reasoning was that having the chaperone machinery in the ER of the mammalian cells will really help with assembling the disulfide bonds correctly. It gives us well-folded disulfide bounded stable protein. To do that, we use this expression construct. So for people who are not that familiar with mammalian expression, we have an N-terminal leader sequence which guides the secretion of the protein. We use a his tag for purification. And then we have the siderocalin fusion, which is siderocalin is one of the most abundantly secreted protein in human cells. And the Olson and Strong lab at the Fred Hutchinson Institute shows that that is a very good protein to attach as a solubilization and secretion tag for mammalian secretion. And then we have a TEF cleavage side, and we have our designs. So in this construct, I did a test expression in a 1 milliliter plate expression culture. And so if we express them at 37 degrees, I only really observed one design that expressed well. And for the other ones, there was no expression visible on this Comasi gel. So if I do a Western blot for the flag tag over here, we can observe some more designs that seem to express visibly on the Western blot. And secondly, on this non-reducing gel, also see these higher bands, which are probably intermolecular disulfides. So the mammalian cells did not help in forming the right disulfides in some cases. Nevertheless, I took the most promising expression designs and I switched the expression temperature to 30 degrees. So the rationale is that at lower temperatures, we might get some better protein folding and can elevate some of possible protein toxicity. And so it's expression at 30. I picked 7 designs, 5 designs expressed, 3 designs expressed pretty well. So I moved into some purification where the immobilized metal affinity chromatography is the first step. And after we bind the protein, we elute an imidazole. And so 3 proteins purified pretty well for that. After a buffer exchange, I do a TEF cleavage to remove the tag from our mini-proteins. And so shown here is the TEF cleavage. And so in all 3 cases, the protein is cleaved off the tag. And then I perform a secondary IMAG where I want to remove the uncleaved protein, which shouldn't have a his tag anymore, from the cleaved protein and the TEF protease, which are both still his tagged. But if I do that, I didn't detect any of my design proteins in the flow through, but they co-eluted in the second IMAG dilution column. So I try to separate that by size exclusion. So here I'm going to show you a size exclusion profile of an analytical column. And so I get 2 peaks. And it seems like that the design is not that easy to see on this gel, but seems to co-elute with the first uncleaved protein. So I never observed any aggregation or anything in my samples. So I just assumed that they might be a bit sticky. And short of adding detergents, we decided to try excipients. So excipients are additives we can add to our buffer to make proteins more soluble or more stable. And so in the literature, we found this excipient buffer, which has arginine, glutamic acid, and isoleucine. And so if I dilute my TEF cleavage protein into this buffer and repeat the secondary IMAG step, now I get my protein in the flow through. So really happy about that. So I got some protein to do some further characterization with. So how do we characterize it? We want to figure out if they are active agonists. And for that, we use a glow sensor. So the glow sensor is this modified firefly luciferase protein that can bind to cyclic AMP or cyclic GMP. And so if CMP or GMP are around, these 2 halves in the N-terminus on the N-terminal part come together, and the luciferase increase in luminescence, and we can measure that. And so we can do that for our family B GPCRs because they are all signaling via the GS proteins. So if they get activated, the GS protein gets activated, that then activates aryl cyclase. That's an enzyme that converts ATP and cyclic AMP. If that happens, we measure the increase in cyclic AMP with the luciferase sensor. So how do we do this assay? We have this HEC cell line that stably expresses the glow sensor, which was made by James, another postdoc in the lab. And so I could take the cell line and then transiently transfect our receptor into it. And after I transfect it into the cells, I can harvest them, assay them in a plate at our peptide in control, and then measure an increase in luminescence to see if there's signaling. So some controls. On the left side now, I show you untransfected cells that don't have a receptor, and on the right, transfected cells that carry the PTH1R receptor. And so in green is a positive control. It is phoscolin. Phoscolin is a direct activator of adenylate cyclase. So it just measures if our glow sensor is active and detects the increase in cyclic AMP when adenylate cyclase is activated. And we expect it to be the case in both cell lines, and that is the case, shown in this green line. In the negative control, the buffer, there's no change in either the untransfected or the transfected cells. And then the natural peptide ligand PTH134, if I add it to the untransfected cells, no change as expected. But if I add it to the transfected cells, there's a significant increase in signal. So I can do a dose response curve where I serial dilute the peptide and measure the signal. And so if I do this, I can get this, I can fit this dose response curve to an equation and estimate an EC50. So, and the EC50 I estimated was 3.5 nanomolar, which is in the range of the published EC50s for that receptor. And the most different seems to be which cell line is used and which assay is used. But overall, I concluded that this assay works as expected in our hands, so we can validate our design with that. So now to the design. So here's now shown the transfected cells. In green is the buffer control. In blue is the PTH native peptide control. And then in red is our design water 24. And it seems it signals to almost the level as the natural peptide. So I'm pretty happy to see that one of the designs is acting as an agonist for the PTH1 receptor. And I can also titrate that down. So if I go to up to like low nanomolar, high nanomolar concentrations, I still get signaling. And also there's no unspecific signaling in the untransfected cell lines. So that's really exciting. And if I just compare that to the native dose response curve, so it's still not as good in this first test, but for being the first generation of these designs, and we, basically from our first 48, getting one decent agonist is pretty exciting, and I think really cool to see. So lastly, what I did is I just looked at the secondary structure. So I'm showing you here a CD spectra. We can see the secondary structure, and alpha helical signals usually have a minima at 222 and 208 nanometers with a maxima at 195. So our water 24 is definitely helical in the CD spectra. So with that, I'm already at my summary. So we can rational design a de novo functional agonist for class B GPCRs, which is very encouraging and exciting to see. We saw that there's some challenges in making these de novo proteins in mammalian cells. So we get these unintended intermolecular disulfide bonds that are forming, which might be cost to the cells, might be something we have to improve in our design protocol. We might have some too much hydrophobic surface, which might lead to the stickiness we saw in the purification. So there's some improvements to be made there. And excitingly, this excipient buffer seemed to work as a really good mild alternative to detergents to help with the not stickiness. And so, yeah, next step, I'm purifying the active domain by itself and measure binding. We are going to put that successful design in a yeast surface display vector to see if we can display it. And then that's a good way to do affinity maturation to improve the binding affinity and make it a better agonist. And then we will take the lessons learned from this design to improve our design protocol. So one idea is to remove the disulfide one step and then introduce them later, so we can maybe test more designs in one go. Make some improvements to the interface design, introducing more hydrophilic surfaces. An idea is to use the HBNet mover for that. And then also maybe adding a fourth helix to just increase the percentage of protein that has a hydrophilic surface to the binding interface. And then I want to convert it into an antagonist by removing the agonist part of the peptide. Yeah, and then, of course, designing the agonist and antagonist for the other 9 targets we have. So with that, I want to acknowledge Chris, has been a great mentor, and Frank, Jared, Paul, and Francis, who really took me in and teach me how to do computational design, which I did not know before. It's really, really fun and awesome. I want to thank James, Ian, and Isabel for helping me with getting up and running in the wet lab, teaching me how to use mammalian cells, helping me with the transfections. I want to thank Sarah, Dan, Edson, Arturo, and everyone for the great atmosphere in the lab. And I want to acknowledge Samina, who previously spearheaded this project. I want to thank the IPI for making it a great work environment. And I also have to thank Frank and Andromeda, which is our supercomputer cluster, which gave us all the CPU hours to do that. And yeah, Frank does a great job building and maintaining it. So with that, thank you for your attention, and hand free to answer any more questions about our pipeline or our designs.
Awesome. Thanks, Ben. It's a really excellent seminar. Yeah, open up for questions.
Hey, Ben.
Hey.
This is really cool. I think it's awesome that you guys got a successful binder and that it does what it's supposed to. I had a couple questions about the process of designing. So I guess you might have already said this, I might have missed it, but in the initial sewing step, when you were sampling backbones, I guess this is a first part of a question. Were there clashes in some of the designed backbones between what you sampled for the structure of the peptide or the addition and the protein? And was that a lot of the designs, or was it not that many? I guess I'm kind of wondering how many things you sampled you had to eliminate right out of the bat just because they clashed.
So the So Anything mover, yes, it actually throws out everything that clashes. So it samples millions of these fragments, and in the end, we throw out all the clashes before we even filter anything. So yeah, there have to be millions of clashes I never see.
Okay,
but
cool.
Just samples it.
Yeah, that makes a lot of sense. I'm also wondering, in terms of diversity of backbones, is there a metric? Do you compare them and see how your diversity grows with the number of ones you sample? And do you feel that was kind of saturating, or maybe you just got enough so you didn't need to look for all of the possible backbones?
So when I make almost 2,500, I was wondering the same thing if I sampled all the possible backbones, but then because there's so much sampling going on to get these, right, we have tens of thousands of backbones that then we filter with some of our criteria to then get down to the 2,500. So there have been many more bad-ish backbones sampled before, and then that number seems good enough to give it a try and see, because I don't think that more sampling, my idea was that by trying them, I will learn more than by sampling more and maybe finding later out other flaws that I didn't predict from just doing the computation experiment. But I think maybe Frank has a bit more feedback on that.
Yeah, I can confirm you're nowhere close to saturation. We do have those metrics, they're unpublished. But when I run them on just random sewing runs, we're nowhere close to saturating at these scales.
Cool, that's really exciting. Okay, I'll let other people ask questions.
Yeah, Ben, I have a quick question. Are you able to rationalize the bad Walters? look back at some of those sequences and use some old-fashioned metrics like number of hydrophobics or hydrophobic surface area, or for all these reasons why they might just not be expressing or insoluble.
Yeah,
Wasn't
so
that
I
the
guess
whole
this is it.
point of Breaking Bad was that Walter needed to rationalize his bad behavior?
yeah, like so the overall goal is to make them into Heisenbergs, right? So if we have then Heisenbergs, that should be the final design. But yeah, so for the hydrophobic metrics, yeah, so I look at the Sasa ratio of the designs, and so that's the spectra they fall in. And so here, like that's also final 48. And so Walter 24 is like kind of in the middle of the one I designed. So it's like, yeah, so all these numbers, we looked at structures from the PDB, and these are ratios that are definitely happen in nature. It's not higher than what you observe in natural proteins. So it's definitely possible that they just get aggregated and sticky and are toxic to the cells for sure. But I don't know if that's a single metric that determines that, or which makes them worse than others. And it's probably by just 48 designs we ordered, right? If some disulfides are not well, and then some others are hydrophobic is maybe, so there's like, it can like, many different designs can fall for one reason, and then with 48, we just haven't sampled enough. So it's really exciting that we get 1 out of 48, right? But yeah.
I have a question along kind of in the similar vein of it's really cool that you got one to work and that it, to purify and that it works. And I know you mentioned that this really only worked once you started including those additives, which as someone like from a protein purification background, you can post hoc rationalize why it worked however you want, but did you not show others because they didn't work or because you haven't gotten to them yet? Because I'm kind of curious, like, is the approach that you want to take now trying to optimize this one hit that you got? Or are you also taking into account that, if you had the protein purified, there could be other better starting points that you haven't been able to find yet? And I guess my question is, how do you balance those of working with what you have versus seeing if you're kind of in like a local minimum?
Yeah, no, that's a very good question. So I shot the one that worked because it's the only one I really tested in the signaling because I got it the best, like to purify. And so for the other ones, so my rationale is that I can improve the design protocol faster and just order a new set of better designed proteins than spend my time on optimizing purification conditions for designs that I can make, but right, that would just take time. And using we learned quite a few lessons from my projects and other projects to optimize what we do. And I think it's just like we decided another time better spent to make a new round of designs. And then for this one, right, knowing that we can display them on yeast and do our selection for affinity maturation is still a very important proof of concept. So we can use it for other designs too. So I'm going forward in doing that while in parallel designing new proteins.
Okay, yeah, that totally makes sense. Thank you.
So you designed you redesigned the interface of the naturally occurring protein, right?
Yes, we designed the interface.
Is there a reason you just didn't maintain the native contacts?
So I guess we allowed all the native residues to be designed too. So we didn't right in the design, we didn't exclude them to be designed. Yeah, and in the end, right, we want to, yeah, and yeah.
Is the
So I'm like,
I can't remember if it's the N terminal or the C terminal.
The C terminal.
So the C terminal that binds in the
active domain.
That doesn't have, like, if you just maintain that the naturally occurring helix, you might actually improve your binding affinity to that of the native, right?
So, okay, sorry, I guess I misunderstood your question. So we maintained the first 16 residues, which is the agonist part. I completely
Okay.
maintained. They are completely the same. So we only designed everything. So everything
Okay.
in my first pictures, everything what was the pink part, which is in the receptor, that's identical. We only changed everything basically 6 after residue 16 and upwards,
Okay.
which is the antagonist part. Yeah. Yeah.
So now maybe you could find mutations that improve your affinity.
Yes.
Down to those 16 residues. Cool.
Yes.
So I have a question on the strategy as well. So I guess you're making another agonist first to prove to yourself that you have your 3 helix bundle and that binds in the right place, I guess, which is clever. And then the ultimate goal is to take away the N-terminal and to have something that binds stronger in that ECD that then generates the antagonist. Is that the strategy?
Yes, that's a strategy. So
and so then couldn't you? So I thought you wanted to. So you said something about adding another helix to, I guess, increase binding affinity upstairs there on the ECD. Could you also think of making a monkey wrench in place of the N-terminal, like, I don't know, a beta sheet, a turn or something that kind of screws up the where the N term is right now?
Yeah, so one idea is to use the Hemlock protocol and make a disulfide bound on the N-terminus after removing the first 16 residues. So you have it disulfide bounded on both ends of the protein. And so that's definitely an idea we are thinking about doing. But first we have to, and then the agonist approach, we choose, as you pointed out correctly, that if we can make the agonist work, then we know that our proteins can bind to the active domain. It doesn't disturb anything. And measuring agonist activity with this cyclic MP assay is very straightforward and easy. So that's what we did there. And the other helix I wanted to add would be more on the back part of the protein to maybe make it a little bit larger, and that might just help with solubility. If we can't solve that with changing the interface residues or other design strategies, then just making it a little bit larger just might help to change the percentage of surface to binding interface. So it's more like not really to make the binding affinity tighter, but just to make it a more soluble mini protein. If that's something we have trouble with still down the road.
Nice. Yeah. So you can basically see what strategy works and then vary around these strategies, basically come up with better call salts or whatever.
Yeah.
All right. Cool. Thanks. It was a really nice talk, I thought.
Thank you.
Maybe you could describe a bit about the So Anything. What did I have to catch up a little bit about how does that differentiate from the previous sewing protocol? You described it being very modular.
I guess Frank is the better person to answer that.
Sure. The main difference is that in classical sewing, the entire backbone design happens in a single step, so there's no chance to do anything else to the protein. You literally put in a helix, you get out a design. The new one actually works with poses throughout, so you can do things like filter in the middle of the design run, or in this case, filter between the design run and the Hemlock stage using conventional Rosetta filters and metrics and so forth. So instead of being this one all-encompassing protocol that barely fits in Rosetta scripts, it's just a standard set of movers.
And Hemlock is also part of Rosetta scripts.
It's basically just our combination of having one So Anything step and then using the disulfide mover, which already exists in Rosetta, but just giving it enough a loop to find a good position to put the C-terminal disulfide.
Mm-hmm.
Cool.
I noticed in your layer, I guess your design restrictions where you define your amino acids that go into the layers, you don't have any aromatics in the core,
So,
which is different than what I went and checked. It's different than what is recommended in the documentation.
So we use, we allow phenylalanines and we allow, we know phenylalanines. So I don't allow any tryptophans and methionines because we don't want to have any residues that can oxidize down the road. We want to really use it for any real therapeutic applications. So we just exclude them.
You left those out completely. Yeah, I see that.
Yeah. So phenylalanine is the only aromatic we allow in the core.
Except for cysteines.
Is that just a quick trick to not have any buried unsats? Okay.
Yeah.
Chris is nodding his head. I've just designed some proteins and I'm having a hard time getting rid of those tyrosines with the buried unsats. So I'm going to go and use that.
Cool. Well, if there are no other questions, thanks again, Ben. Really enjoyed the seminar.